Stephen Helliwell is from Rejuveron, another very exciting organization that doesn't fit into a neat biotech company box. Lots of exciting things going on, some pipeline in clinical, is that right? So interested to know how things are going, Stephen, so over to you.
Great, thank you very much. So it's very nice to be here today. Thank you very much for having me. One of the disadvantages of going midway in the afternoon is that people are starting to sleep after their lunch. One of the advantages is, of course, that many people have laid down some of the sort of concepts that I'm going to, be able to skip over briefly because they've already been mentioned. I was particularly pleased when Krista mentioned the work of yeast scientists in the 90s in discovering the whole longevity field, because I actually did my PhD with Mike Cole when we discovered the target of rapamycin TOR. Full disclosure, though, we did not do any longevity experiments. We left that to Brian Kennedy and others later on. So Rejuveron is based in Zurich in Switzerland. It's a platform biotechnology company. We raise funds like a biotech company, and we deploy those funds to incubate early stage subsidiaries, or we invest in already existing companies that we're very excited about, regardless of their stage in actuality. And so we have sort of this mixed mode model. And we currently have 5 companies in the portfolio. And by the end of the talk, you will know a little bit about all of them, I hope. So we have a vision not unlike other people's. Our motto is age healthy, live longer. Age better, live longer. Obviously, we're trying to push the health span. If we compress morbidity, it's good. If we don't compress morbidity, people will also live longer by definition. So we're also working on longevity, but we are aiming to improve health span. Very briefly, you're not allowed to come to these conferences unless you show the hallmarks of aging. So I put it in as well with our graphic design. The hallmarks of aging are an excellent biological framework within which to talk to non-scientists about aging, because this is very complicated. But in reality, we can be guided by the hallmarks. But if there's a piece of aging biology that we've identified from an academic or wherever that we want to invest in or start a company with, it doesn't have to be in the 9 hallmarks of aging, just to be clear, right? And these change all the time. They're dynamic. It's been 6, 8, 9, 14, 11, depending on what you read. So why are we unique from everyone else that you've heard from today and other people in the field? It's in reality, the number one risk factor for many, many diseases is aging. You've also heard that before today. So we aim to develop novel therapies for age-related diseases or for diseases where the biological mechanisms are related to aging. Okay, I'll come back to that point in a minute. When we look at a particular opportunity to start or incubate or invest in, we want to see a very clear clinical path or a concept for a clinical path to reach a phase 2A proof of concept trial, because this is the moment when you have your biggest inflection point. This is the moment you know that your drug is safe for a certain amount of time, but is efficacious in that first indication that you've chosen to prove that your drug is working. And this is really, really critical for the company and for the investors that are expecting a return, such as yourselves. We also plan to aim at chronic diseases or age-related diseases with a clear unmet need, because if you move into a very tough space, you're going to have a tough time. So low levels of competition in a particular disease area is of advantage. The overarching vision, if you like, is to take these molecules into unmet need situations, but to try and immediately expand into indications of ever decreasing severity and eventually to end up in otherwise healthy aging populations. So although we're modality agnostic, we tend to focus more on small molecules or simple biologics like antibodies, modalities that have been well validated in the clinic, because scaling cell therapies and organelle therapies and so on to hundreds of millions of people one day is still not practical. We do this all in Rejuveron in Zurich. We have laboratories in Zurich. We have lots of scientists there, incubator space. We diversify across stage, hallmarks, modality, I've already mentioned that. And we allow scientific founders to come in, incentivize themselves with founding shares, etc., and we guide them with our clinical expertise to success in the clinic. Very briefly, the team. You've heard Aaron this morning giving an excellent high-level sort of view of the investment arena, the current environment in the space. And of course, he works with Christian Angermeyer, who is our major funder and founder. And we have an excellent management team, many years of pharma and biotech experience. Not on this slide is our lead chemist, Patrick, who has 22 years of experience at Roche and put 7 molecules into the clinic. So we would like to think that we do know what we're doing a little bit. I'm just going to tell you now very briefly about our 5 portfolio companies. So we have 5. Endogena, RejuvenateBiomed. These are 2 companies that we are invested in. They're more or less standalone companies with a fully functioning team, and they're both in the clinic. With 3 companies that we're more incubated based on what we've started for ourselves are Rejuveron Senescent Therapeutics, Telomere Therapeutics, and Vascular Therapeutics. So I'm just going to take you through each of the 5, and then I'll be very happy to take your questions about any or all of the 5. So endogena. Endogena works on retinitis pigmentosa, which is a disease which gradually kills off the rod and cone cells in the back of your eye, in the back of your retina, based on various mutations that you receive at birth, but there are also some environmental factors. And there are stem cells at the back of the eye as well. They're endogenous stem cells. They're there all your life. And so endogenous found a way to take a small molecule, which actually is injected intravitreally. So it's injected into the eye. So it's local delivery. It's very good clearance in the rest of the body. So we've got no safety worries in that sense because it's cleared very fast systemically. And this molecule enables the triggering of the division of these stem cells more, and those stem cells can then differentiate back into rod and cone cells of the outside the outer nuclear layer in the back of your eye, preventing vision loss occurring at the same rate as it would otherwise have done. Endogena are in a phase 1/2A rising dose trial, primarily for safety, but they are reading out relevant clinical endpoints for visual function, response to light, and so on. So we're well on the way here. We're expecting, we're hoping for exciting good news about results, and we're going to get readout on this trial beginning of the 3rd quarter next year. So it'll be a major news splash for Rejuveron and Endogena. And at Endogena, there's a second program that's also focusing on the eyes, focusing on geographic atrophy, which is when regions at the back of your eye sort of get damaged and die out. And that's obviously part of the phenotype that causes macular degeneration, which is also, like retinitis pigmentosa, a disease with entirely unmet medical need. There are no approved therapies for either, for one form of AMD and all forms of RP. RejuvenateBiomed, when I first joined Rejuveron, one of the first things I had to do was go looking for more companies to invest in. And I came across Anne Bellion in Belgium running her company, RejuvenateBiomed. Anne has a strategy, if you like, not unlike what Kristen has just talked about, using known drugs that already work and finding age-related indications to work with. Anne went one step further and came back to a theory that someone else said today about combining drugs. We shouldn't do one drug, one target, etc. Combining drugs together to attack multiple pathways that go wrong and cause aging. So Anne's company's first molecule is a combination of 2 approved drugs, each of which have been in tens or hundreds of millions of patients already with very, very low side effects, very low safety features. And the combination of these 2 drugs in mouse models of sarcopenia form an incredible effect on the ability of 2 different mouse models to keep their mobility and their motion. So the graph on the right-hand side of this slide shows drug A on its own, drug B on its own, versus untreated, almost no effect on, in this case, running time. We have lots of other data here. And when you combine these 2 drugs together at the same dose as A and B separately, you have this remarkable effect. And this is a mouse model that matches a sort of semi-acute, semi-chronic mouse model of sarcopenia. And as you've just heard from Kristen, age-dependent sarcopenia is also a fully unmet need. Some people's muscle declines more than others as they age, but if these drugs are really as safe as we think they are, we know they are, but if they're also safe in combination, this is the kind of drug that you could see being given to everybody over a certain age, simply to maintain their muscle mass. So this is super, super exciting in terms of the expansion potential is enormous. I should say there, I've already said it once, we're in the clinical phase 1B. We are in a casting clinical trial. So Kristen talked about casting in her mouse models. We're actually casting over 65-year-old humans in Belgium. They're having a 2-week whole leg cast on one leg. And while they go on the drug, they maintain the drug for another 2 weeks, and then they get readouts over time for the return or the retention of the muscle mass and function that they've lost during the casting. Okay, that will also read out in the 3rd quarter of next year. So on to the 3 companies where, if you like, they're slightly sort of more internal, ones where we have a much more direct role in how they run today, in terms of myself and Patrick and others on the top team at Rejuveron. First one is Senescent Therapeutics. Of all the aging-related sort of features of biology that is sort of the most advanced in this very early nascent field, it is the concept that if you kill the senescent cells in your body, you are going to do better off in life. And so one of those areas that's sort of a specific area, this is a spin-off out of Manuel Serrano's lab in Barcelona. He's a very famous oncology senescent scientist who's now also at Altos, which is also good for us. Reflected glory is also good. And so Manuel had discovered that when you treat certain solid tumors, there's an occurrence of senescent cells within the tumor. So these cells have escaped the cell cycle, become senescent, then they can't be killed by cell cycle acting chemotherapy drugs. They tell their neighboring cells to be senescent and keep a few cancer cells alive at the same time, and the cancer regrows and never goes away. There's also an effect of these cells on metastasis of these cancers. If you treat either subsequently or in combination with our monoclonal antibody therapy against a cell surface target, which is called PDL2, expressed on these blue senescent cells on the left, you kill all the senescent cells and you kill all the tumor cells. There's nothing left to regrow. Okay? So on the right-hand side, you'll see a graphic. This is actually from Manuel's preprint in bioRxiv, so last month. And if you see they're not very good colors, are they? If you see kind of the olive green color here, these are mice carrying triple-negative breast cancer cells in a tumor on their flank. It's a very normal standard mouse model. And if you see the olive line here, there's basically a complete ablation of this tumor when you combine our anti-PDL2 antibody with the classic standard of care in the mouse model, which is doxycycline here. Okay? If you use either agent alone, you basically have no effect as compared to vehicle control. So the combination is remarkable at completely ablating tumors. So we're very excited about this. This will go into the clinic just before the end of the year, next year. So Q4 23, maybe Q1 24. But it's coming along very, very well. Telma Therapeutics. So our co-founders, who shall remain nameless, are on the east coast of the States. They discovered a novel gene that when you knock it out, can reverse the telomere shortening of several different diseases, genetic diseases, that cause premature telomere shortening. So you'll all know that everyone would like to keep their telomeres as long as possible in their stem cells in their body. There are various ways to do this, some carrying more risk, some carrying less. In our particular case, we already know that the target that he knocked out, on the right-hand side of this slide here, you see in the 2 pink boxes, these are patient stem cells with disease mutations. So the length of their telomere is shorter, bottom of the gel. A wild-type cell from an age-matched person is the grey box on the left of the panel, and the blue box is the knockout of the gene that reverses this telomere phenotype. So on the far right of the gel, you see the telomeres have gone back to normal size, but they didn't get longer. Okay, so not getting longer is good if you're worried about cancer, because that's how cancers can occur. Okay, we know that there are compounds that have been in humans for other diseases, entirely unrelated, that also hit this target. So all we have to do now is make novel small molecules that are more selective for this target based on what's been in humans before. And we're well on the way to doing this. We've got some excellent chemistry going on. And we're still, I would say, 2.5 years from the clinic, but we absolutely understand the mechanism. This mechanism is related to the biology of aging. And this will be a pediatric trial. So not a trial in aging patients, but in pediatric cases carrying these mutations. The hope is that we can then expand to other diseases, such as idiopathic pulmonary fibrosis, where the AT2 alveolar cells in your lungs also extend as a over-shortening of telomeres during the etiology of IPF. And eventually we would hope to also see if we could go into healthy people one day and keep their telomeres longer. Last but not least of the 5, Vascular Therapeutics. This is a spin-off from Professor Lee Rubin's lab at Harvard in the Stem Cell Institute at Harvard. You may have heard of experiments where people hook old mice to young mice via their blood supply, so-called parabiosis, and the old mice exhibit features of rejuvenation by being connected to these younger mice. So there's various offshoots and outputs of spin-offs and startups based on these observations. But Lee Rubin has single-cell data from brain endothelial cells and other cell types in the brain, showing what's changed again in those brain endothelial cells in terms of the transcriptome to make the old mice appear young again. Okay, so the plot on the right is simply one of the plots of the genes that change in old mice brains versus old mice that's been connected to a young mouse and exhibits rejuvenated phenotypes. It's a bit gratuitous, don't worry about it. The point is that we're identifying the pathways that make these genes change their expression and working out which ones are causal or not. And based on this, we built a chemical library using AI, machine learning technique, to make a very small library. We ran it over an assay for brain endothelial cell function, and we had a very high hit rate. So our AI engine to make a chemistry library was very good, and we're now we're working on the lead up of one of those molecules, actually against quite a well-known target, but we found a way to think about going into mouse models of small vessel disease. So small vessel disease is a disease where your blood-brain barrier declines over time in all of us as we age, or by certain insults. So American footballers have traumatic brain injury, they have small vessel disease, but Alzheimer's, dementias, various other brain-related indications all have elements of small vessel disease as part of their etiology. And this is incredibly understudied. The retention of the integrity of your blood-brain barrier to stop dirt from your blood getting in your brain and causing neurological inflammation is an enormous big deal, and no one is doing it. So it's the most exploratory company we have, but it really has the potential to have a small molecule that improves blood-brain barrier function. As you age, it retains or improves blood-brain barrier function and could prevent dementia and other diseases which are absolutely untouchable currently. So thank you very much. There's a summary there which you can read as I kind of sign off. We're a biotech company. We have a portfolio biotech company. It's decentralized. We have people in Zurich, we have people in Barcelona, we have people in Toronto working in our companies. We have 2 assets in clinical trials, and we have, as I've already said, excellent facilities in Zurich, which you're very welcome to come and visit, and I hope to see you there.
Thanks very much, Stephen. Great, and we're now, thank you.